So today I'll be talking about a paper called Self-Play Reinforcement Learning Guides Protein Engineering. I think this paper was pretty cool. And the reason why is it has something closely to do with like the two most exciting pieces of news out of science, I think, in the recent years. The one on the left is basically a New York Times covering the fact that computational protein design has managed to um, create a protein that seems to replicate the abilities of CRISPR, which is really exciting. And the one on the right, I think people might remember from a few years ago how a reinforcement learning algorithm basically beat one of the best human players in the game Go, which is incredibly complex. And today, hopefully, I can show you how this paper tries to bring these two efforts together and then point towards a new direction for protein design. So you've heard a few protein design papers in this class. And I thought before I go into this paper, I would like try to recontextualize what protein design is trying to do, and also to show why we want to talk about this. Right. So the problem of de novo protein design is to design a protein that is different from what's occurring in nature for a specific function. So some of these functions might be like binders, where you have like T cell and the CAR T, and other things that people might be interested in is, for example, optimizing for like the fluorescence of a GFP, right? So let's suppose you get a protein and somehow, and then you can run some assays to validate it, right? But the problem is that while we usually think about protein function as being like some sort of like 3D structural property, the way we commonly like then produce this engineered proteins is in 2Ds, right? So we insert a sequence into an expression system, and then the system kind of like expresses the protein, does a bunch of processing, and then you get a 3D protein in solution. That's what usually happens. So then you have to do this additional step of like, after you design the 3D protein, you have to somehow like create the 2D sequence. And that's not exactly a trivial task. So we've heard a bunch of approaches in this class. Hoyan told you about RF diffusion, which uses a diffusion-based model to generate protein-like 3D designs, and then use something called the protein MPNN to refold, kind of inverse fold that back to a 2D sequence. John talked a little bit about how to use AlphaFold metrics to then validate that this 2D sequence is in fact designing for the thing you want, and to increase your success rate. And we've also heard about a yeast-based expression system where you can test the experimental protein-protein binding interactions. So why do we need a new paper? It seems that this area is like well covered, right? Well, not quite. So far, all the approaches you've heard follows this kind of template. You start off by designing a plausible looking protein. That's essentially what the diffusion model is trained to do, to like denoise like a bunch of Gaussian random noise to a protein that looks like something in the protein database. And you can try to constrain that process using geometry or motifs or binding partners. But essentially, you're still looking for a plausible protein, not something that's targeted towards the specific function that you want. You can validate the plausible protein to eliminate a bunch of candidates, and then you inverse fold it back to sequence. And then you have to refold the sequence back to structure using something like AlphaFold to check that you haven't lost anything during this process, right? And currently, like the state-of-the-art success rate is still something around single digit. So you have to keep pulling this lever like hundreds and thousands of times until you experimentally find a successful candidate. And this process is incredibly expensive, right? And more importantly, once you succeed, you can't really inform your next design. You just keep on pulling this blind lever. So what we would like to do is to learn a targeted approach. So that's what this paper tries to do. In red, you have all the parts that we've talked about previously in the class. And the paper kind of like boxes it away and calls it a surrogate model, where you have something that tells you maybe how a protein sequence in 2D is supposed to function. And this is a useful abstraction, right? Because then you can use this method on top of any like state-of-the-art ML models. And what this method is trying to learn is this thing in blue, which is the policy value network. And it's a network that evaluates a protein sequence and tries to give you like the optimal decision on how to design it or how to mutate it so that you reach your best target. So before we go to the protein itself, I just want to talk about briefly the method that's underlying this paper. And it's a method called Monte Carlo tree search. And essentially, Monte Carlo tree search is born out of kind of reinforcement learning world where you imagine you're playing a game like chess, right? So in this tree, the white nodes are a move that the white player makes, and then the dark nodes are the move that the black player makes. And Monte Carlo tree search essentially simulates playing many, many games and looking at the successful moves from the past and then expanding upon those successful moves in the second column. And then further simulating self-play until you reach some sort of like win-lose condition, and then updating this tree. So what you're doing is doing this like probability weighted walk of potential plays. And the idea is that you can then find moves that win most often and then play those moves more often. So the paper kind of ports this idea over to the protein design world, where you imagine the game as designing a sequence of proteins. So each base or each amino acid, there's 20 possibilities. And it's important to appreciate that the solution space is huge, right? Like it's 2 to the power of L. And if L is like 100 amino acid protein, this is like more than the number of atoms in the universe, supposedly. And to traverse this like incredibly complex design space, the authors frame this problem furthermore as a single point mutation of this amino acid chain. So you start off with some random sequence, and at every move, you can change an arbitrary base, an arbitrary position to an arbitrary amino acid, and you keep doing this in hopes of like searching for the optimal protein of this defined length. So you kind of go through the same Monte Carlo tree search process where you iteratively update each amino acid. And eventually, and before making a mutation, you want to simulate the downstream consequences to some depth and then choose the best mutation, right? And then you keep running this until your computational budget is spent. And eventually, you will end up with not only the optimal sequence given your design budget, but you also end up with this like optimized policy value network. And this is the main benefit of this method, right? Like the next time you want to redesign something of this sequence length for this particular function, you don't have to start from scratch. You start from your current best understanding of how to optimize within this sequence length to a particular function. So they performed a couple of benchmarks. The first one they did is a simulated benchmark where they try to optimize GFP fluorescence intensity. And they're designing over a protein sequence of a like 237 amino acid length. And in the simulated setting, they just use an ML model to approximate the true fluorescence intensity. And the goal here is just to show that the method is capable of optimizing over some design metric. All right, so indeed here, they show that their method, which is in blue and purple, outperforms a bunch of baselines using the same computational budget. And that's important. And they also suggest that their method achieves a good trade-off between generating diversity and function. So in terms of generating good function, their method, which is in the top row, is able to generate more hits of like mutations where they consider the fitness as good over a bunch of initializations compared to benchmark. And then in terms of diversity, they show that their method covers more of this dimensionally reduced space without compromising on the ability to generate good hits. So, okay, so they can do well in simulations. The next thing we want to do is to show that they can actually design good, well-functioning protein. And in this case, they choose a slightly different protein, which is the luciferase, and they want to optimize for the bioluminescence. One important thing to note is that it's incredibly hard to perform experiments over this large search space. So they reduce their search space down to a smaller subset of bases of the protein, which they derive through looking at commonly mutated bases as well as known bases with functional rows. So they started off with a pool of 164 experimentally constructed and validated variants. to seed the model as a training set. And then they train this model to generate an additional 1,500 variants across 10 independent runs. From this 1,500 variants, they performed deduplication because it is possible across independent runs for the model to generate the same candidate. And then they performed k-means clustering to find 36 canonical model-generated variants, and out of which they achieved a pretty good success rate where they could express a majority of the model's variants. And 11 out of 36 of the model's variants outperformed anything within their library of constructs. So this shows that the model is indeed like optimizing on some axis. And you can see that in the plot on the right, that the best candidate that the model generated is like far more luminescent than the best candidate in their library. So they've showed that they can optimize over protein design for a particular property, but people are also interested in designing proteins to interact with other proteins. So in this case, they design a protein-protein binder to bind with a ribonuclease. So in the diagram on the right, you have the ribonuclease shown in cyan and the binder that they are designing in this pink magenta color. And during training, they try to approximate this binding task with a AlphaFold derived accuracy metric. So the first thing that they showed is that their method is able to achieve similar performance to benchmark in terms of optimizing on the AlphaFold metric. But very importantly, they're using a much less refined version of AlphaFold. So for those who know how AlphaFold works, you iteratively feed a sequence through the model multiple times for refinement. And that is computationally really expensive. So by using something which is less refined, they're able to like search over a larger space given the same time constraint. In addition, the top binder that they designed folds stably during molecular dynamic simulations. So on the right, you have the RMSD of the different atomic coordinates, as well as the number of hydrogen bonds in simulation. And what you want is a relatively stable kind of curve to show that the protein is indeed a stable candidate. And they achieve that. So lastly, what they do is an experimental validation that binding is indeed occurring. And they achieve this through the biolayer interferometry assay, where you have basically a very sensitive kind of like force measurement and one protein on one plate and another protein on another plate, and allowing them to kind of bind and dissociate so you get a binding curve. This binding curve is not like intuitive to look at the first time, but what you can appreciate is that for all the proteins, there's an increase during association phase and then a fall during the dissociation phase. And this curve allows them to calculate this KD, which is a binding constant, and the smaller, the better. And they designed two candidates which are a stronger binder than the wild type ribonuclease target, even though the model has not seen the ribonuclease target during training. So just to wrap up, the strength of this paper is that there's a clear problem motivation in terms of having a directed approach to protein design. They use relatively simple methods with no like fishy loss functions. So everything is well motivated. And they also use strong modern benchmarks, which is something to appreciate in computational papers. And they perform a variety of in silico and biological validation to show that their method is working. The weaknesses is that there is relatively modest performance improvements over baseline. And I think some of this has to do with the data constraint that they have, because it's pretty expensive still to generate experimental training sets to optimize the reinforcement learning agent. At times, the writing is somewhat unclear on the details of how their simulation is run. And unfortunately, the field has moved on a lot since 2024. So more sophisticated methods have emerged. So just to kind of go back to this like protein folding workflow, there's now approaches to apply this Monte Carlo tree search directly onto the 3D structure of the protein, which you might argue it's a better approximation of like functional optimization. And there's a recent interesting work where there's preference optimization built directly into the diffusion model. So you don't even need to have a separate model for protein design. You can use the diffusion model directly with preference optimization to design for a specific property. And last but not least, why now? I think this is really cool because reinforcement learning is known to be data hungry, and this is the reason why it hasn't been used so far. But there's very, very recent advances in all of these machine learning methods that makes predictions better and faster, which makes the training of reinforcement learning possible. So I'm personally very excited for this. Yep, that's it. And any questions?
For the audience, I think we can hear you if anyone talks.
This is maybe too orthogonal, but do you know if they addressed in the paper at all how this stacks up resource-wise against something more experimental along the lines of like a direct, like the best directed evolution strategies? Because if you're already going to the trouble of, you have like designing for some sort of esoteric function beyond, just making GFP better, is it that much? It depends, the starting situation of the lab, but does it outperform stuff like that, or is it better to just invest in, phage display or whatever, what have you, bench side?
Yeah, that's a good question. And I think here the kind of the main gain in terms of like doing all this additional computational work instead of just having a sequence that you're experimentally optimizing over is that you also learn this like policy value function on the side. And basically you get on top of it an estimator of like how good any sequence is that your experiment hasn't covered. And you're not only optimizing over a particular sequence for a function, you're also optimizing this estimator that you can keep using down the line. So I think that's kind of the main benefit of doing this versus just doing experiments.
Question about just in general, like applying reinforcement learning in this field in general, because obviously for reinforcement learning, the rewards function is probably the single most important thing for it to work well. And in this case, a lot of time in this scenario, for example, you're making protein binder, then there's a clear things to optimize over, like binding affinity. But in a lot of like the design scenario, you don't actually know what like you're trying to optimize for. So I wonder if like the authors of the paper also discussed a little bit about like how like whether this approach could be like how you might be able to use this approach in those scenarios where you're not necessarily optimizing for binding affinity, for example, but some other functionality that might not be as well described.
Yeah, so unfortunately, they didn't make a comment here. And I think they kind of like wisely chose to stay with problems where people have like well agreed like consensus metrics of what's good. So for example, like GFP, like the brighter, the better, right? And binders, like the stronger, the better. I do think it's an issue, like how to design the best reward functions. Although like, I guess in terms of alternatives, if you don't have a very specific kind of metric, on what you're designing for, whether you use reinforcement learning or not, there's going to be like this question of like, when do you stop, right? Like, what is the best candidate? So I think like in general, what you want to do is to have like a well-designed kind of functional assay down the line where you're like, this is what I need to prove that I've reached the right design. And then you somehow like extract metrics from that functional assay and then feed it back to RL.
Question. So with this tool, could this be used for affinity maturation if we have a known structure? Because there are many structures available, so we could just skip the modeling of the structure and we could just dive into affinity maturation, which I guess would help design better libraries for phage or yeast display. So is there is that like a promising area with this tool?
Yeah, so in theory, you can do that. I don't know like exactly how affinity maturation work, but I'm like assuming that you're trying to basically like optimize for some like binding affinity metric. Okay. Yeah, in theory, you can do it directly from sequence to readout. The trouble is that the protein space is really complex, and kind of you not only need to learn an approximator, and the reinforcement learning helps you sample the right measurements to get there, but you also need to parameterize the correct approximator. So in this case, like maybe some sort of neural network need to directly take sequence to readout. And I think people have done that, but also people have found that like it's helpful to have structural information for that kind of estimates. So yeah, it's hard to say. I guess in theory, if you have like an infinitely large neural network, you can do it. But in practice, it might be useful to have like structural information as of right now.
Thank you.
Yeah, or one question, is there anything, is there any way this would, there's something in DNA that's like non-mirth, things that you never see? Did anything in this protein space show things like that are impossible, or I didn't really see much of that in the paper, but I'm wondering if these large search models could find things that are just like impossible, or supposedly impossible.
Yeah, that's a cool idea. They didn't go too much in detail for that, but they did like manage to design some stuff that they couldn't express over here. Maybe that's something worth looking into.
Yeah, it's like something where like Earth's evolutionary pressure has not made it possible yet, or something like that. So overall, we do some quick summaries. It was, I think, the history of protein design and the scale of the problem, like in some of these slides, was it good to kind of highlight the need for the large-scale compute, and also how you built up some of the slides and went back to them as well, like the mathematics and the design, sort of qualities. That was a good layout. I think, at the end you commented how the field had moved on, so that was a really interesting comment, and you could even, for some critiques, you could add in one or two other papers and then point to them when you say the field's moved on, so it's good to give a good example. And some of the slides are a little dense, but mostly a pretty good layout, but I think overall very good, and thanks.
Great, thanks.